Right now, as you read this, genes that don't belong to Homo sapiens are working inside your cells, making proteins, fighting infections, deciding whether you'll have a severe reaction to a virus or barely notice it. These genes came from people who died out 30,000 years ago, people who are classified as extinct in every biology textbook, people who left behind almost no trace in the archaeological record except for scattered bones and stone tools, and these fragments of DNA still humming along in your body, still functional, still shaping whether you get sick or stay healthy when a new pathogen sweeps through your city. If you have European or Asian ancestry, somewhere between 1 and 4% of your genome is Neanderthal, which sounds like a trivial amount until you realize that's tens of millions of base pairs, thousands of gene variants, and many of them are sitting right in the middle of your immune system, the complex molecular army that stands between you and every virus, bacterium, and parasite trying to use your body as a home. If you're using this to fall asleep, let your mind settle on this strange fact. You are not purely one species. You are a mosaic, a hybrid, carrying genetic material from at least 2 different branches of the human family tree that separated 600,000 years ago and evolved independently on different continents before meeting again in circumstances we can only partly reconstruct. The Neanderthals are called extinct, and in one sense they are. There are no living Neanderthals walking around, no communities that preserve Neanderthal culture or language or identity. But in another sense, they are still here, distributed across billions of people, still breathing through your lungs when you catch a cold, still producing proteins in your blood when your immune system ramps up to fight an infection, still present in the most intimate and fundamental processes of your biology, invisible unless you look at the DNA itself and see the sequences that are unmistakably, undeniably from another species. The discovery of this Neanderthal legacy inside living people came as a shock to science when the first ancient DNA was successfully extracted and sequenced in the 2000s. For most of the 20th century, the consensus view was that Neanderthals and modern humans did not interbreed, that they were separate species, or at least separate enough that reproduction between them either didn't happen or produced infertile offspring like mules, leaving no genetic legacy. The evidence seemed clear. Neanderthals disappeared from the fossil record around 40,000 years ago, right around the time that anatomically modern humans were spreading across Europe, and the most straightforward explanation was competition and replacement. Modern humans were smarter, more adaptable, better at language and tool use and social organization, and they simply outcompeted the Neanderthals, driving them to extinction through a slow process of territorial expansion and resource monopolization. It was a clean narrative, easy to teach, easy to understand, and it fit the older ideas about progress and advancement that had long shaped thinking about human evolution. Then, in 2010, the first high-quality Neanderthal genome was published, painstakingly assembled from tiny fragments of ancient DNA extracted from bones found in a Croatian cave, and the clean narrative collapsed. When researchers compared that Neanderthal genome to the genomes of living people from around the world, they found something unexpected. People of European and Asian descent carried small but unmistakable segments of Neanderthal DNA, scattered throughout their chromosomes. People of purely African descent, whose ancestors never left the continent and never encountered Neanderthals, carried none. The pattern was clear and could only be explained one way. Sometime between 50 and 60,000 years ago, as modern humans expanded out of Africa into the Middle East and beyond, they met Neanderthals, and some of them had children together, children who survived and thrived and passed their mixed ancestry forward through all the generations that followed. The interbreeding was probably not frequent, probably not the dominant pattern, but it happened enough times that Neanderthal genes entered the modern human gene pool and spread. And today, tens of thousands of years later, those genes are still there, still active, still part of what makes you, you. But here's where the story gets even stranger, where the simple fact of interbreeding gives way to a deeper mystery about what those Neanderthal genes are actually doing and why they persisted. If 2 species interbreed and produce hybrid offspring, you would expect the genetic contribution from the extinct species to gradually dilute over time, thinning out generation by generation as it gets mixed and remixed with the much larger modern human gene pool. 50,000 years is roughly 2,000 generations, and with that much time and that much mixing, you might expect Neanderthal DNA to be randomly scattered across the genome at uniformly low levels, a kind of background noise, genetic fossils that don't do much but haven't quite disappeared either. Instead, when researchers mapped where Neanderthal DNA actually appears in modern genomes, they found something completely different. The distribution is not random. There are deserts, large regions of the genome where Neanderthal DNA is almost completely absent, as if it was actively scrubbed out by natural selection. And there are peaks, regions where Neanderthal DNA is surprisingly common, sometimes present in more than half of all people sampled, as if those particular sequences were not just tolerated but actively favored, spread through populations by natural selection because they provided real measurable advantages. The deserts appear in predictable places, near genes involved in reproduction, sperm production, testis development, and near genes involved in brain development and cognitive function. This suggests that Neanderthal variants in those regions were incompatible with modern human biology, caused problems with fertility or development, and were gradually eliminated over thousands of generations as individuals carrying them left fewer descendants. Hybrid incompatibility is common in biology, a sign that two lineages have been separated long enough that their genomes have drifted apart in ways that don't play well together, and the fact that we see these deserts tells us that Neanderthals and modern humans were right at the edge of being able to produce fully viable offspring. Most of the Neanderthal genome was neutral or slightly harmful in a modern human context, and it was slowly, quietly removed by natural selection, leaving behind only the pieces that were useful, or at least harmless enough to escape notice. But the peaks, the regions where Neanderthal DNA is unusually common and has clearly been preserved rather than eliminated, those peaks cluster overwhelmingly in one part of the genome, one functional category of genes that stands out like a beacon in every population genetic study that has looked at this question. They cluster in the immune system, in the genes that recognize pathogens, coordinate inflammatory responses, produce antiviral proteins, and regulate the complex balance between fighting infections and avoiding autoimmune damage. Genes involved in innate immunity, the fast, non-specific first line of defense against invaders. Genes involved in adaptive immunity, the slower, more targeted system that produces antibodies and remembers past infections. Genes that control how strongly your body reacts when a virus enters your bloodstream. Genes that determine whether a bacterial infection triggers a mild fever or a life-threatening cytokine storm. Genes that shape your risk for allergies, autoimmune diseases, and severe responses to novel pathogens. These are not random genes, not junk DNA left over from ancient interbreeding. These are some of the most important, most heavily selected, most functionally critical genes in the entire human genome. And in people of European and Asian descent, a significant fraction of the variation in these genes comes not from our own species, but from Neanderthals. The explanation for this pattern is both elegant and unsettling. When modern humans first left Africa and entered Eurasia, they walked into a new disease environment, a landscape of pathogens they had never encountered before, bacteria and viruses and parasites that had been co-evolving with Neanderthals for hundreds of thousands of years, but were completely novel to humans fresh from the African savannas and forests. The modern human immune system, shaped by the specific pathogens of Africa, was not prepared for the microbial challenges of Ice Age Europe and Asia. People got sick. People died. Entire migrating bands might have been decimated by diseases they had no resistance to, and the expansion into Eurasia could have stalled or failed, if not for one crucial advantage. Some of the early migrants, the ones who had Neanderthal ancestry, carried immune genes that had already been tested against those same Eurasian pathogens for 300,000 years, genes that encoded recognition proteins fine-tuned to the bacterial surface molecules common in European soils, antiviral systems calibrated to the RNA viruses circulating in northern climates. inflammatory responses optimized for the balance between fighting cold-adapted parasites and conserving energy in freezing winters. Those hybrid individuals survived infections that killed their purely modern human neighbors. They had more children. Those children inherited the protective Neanderthal immune variants, and over dozens of generations, those variants spread through the population, not because anyone understood genetics or made deliberate choices, but because the simple arithmetic of who survived and who didn't quietly favored anyone carrying genetic defenses borrowed from the species that had already learned, through brutal natural selection, how to survive in the very environments modern humans were just beginning to colonize. This is adaptive introgression, the transfer of beneficial genes from one species to another through hybridization, and it is one of the most powerful and underappreciated forces in evolution. We tend to think of species as separate, of evolution as happening within isolated populations, but the real history of life is messier, full of mixing and borrowing and theft, with genes moving between lineages whenever the opportunity arises and the result is useful. The Neanderthal contribution to the modern human immune system is one of the clearest examples ever documented, a case where we can trace the source of specific genetic variants, measure their functional effects, and see directly how they continue to shape health and disease in living populations. Every time you get a cold and your body fights it off, every time you get a cut and it heals without infection, every time your immune system quietly neutralizes a pathogen, before you even notice symptoms, there is a chance that part of the machinery doing that work is built from instructions written not in Africa by your Homo sapiens ancestors, but in Europe by Neanderthals, tested and refined through ice ages and plagues and the slow grind of survival, and passed to you through a chain of hybrid ancestors stretching back 50,000 years to encounters in caves and river valleys that no one recorded and that we are only now beginning to understand through the evidence written in genes. To understand how Neanderthal genes ended up controlling your immune response, we need to go back to a world where your species was not the only kind of human walking the earth, back to a time between 50 and 60,000 years ago when the ice sheets were advancing and retreating across the northern hemisphere, when sea levels were lower and land bridges connected continents that are now separated by water, when our ancestors, anatomically modern humans who had evolved in Africa over the previous 200,000 years, finally gathered enough numbers and enough knowledge to push beyond the familiar landscapes of their origin and venture into territories that were already occupied. They walked north and east, following rivers and coastlines, moving through the Middle East into the grasslands and forests of Europe and Asia. And when they arrived, they were not alone. The caves were already inhabited. The good hunting grounds were already claimed. The valleys and hillsides already showed signs of fire and tool-making and organized groups. Another kind of human was there first, and these people had been there not for decades or centuries, but for hundreds of thousands of years, long enough that they were no longer the same species that had left Africa in the distant past, long enough that isolation and different environments and the relentless pressure of natural selection had reshaped them into something distinct. The Neanderthals had diverged from the ancestors of modern humans roughly 600,000 years ago, a split deep enough in evolutionary time that the two lineages had accumulated substantial differences, but shallow enough that they remained biologically compatible, capable of producing offspring when they encountered each other again after all those millennia of separate development. During that long separation, Neanderthals had adapted to life in Ice Age Eurasia, to cold climates and seasonal extremes, to forests of pine and oak and open tundra where woolly mammoths and reindeer roamed. Their bodies reflected those adaptations. They were shorter and more powerfully built than modern humans, with barrel chests that housed large lungs for breathing cold air, thick bones that could withstand tremendous physical stress, and heavy brow ridges over eyes that might have been adapted to the long twilight of northern latitudes. Their brains were as large as ours, sometimes larger in raw volume, though shaped differently, with more development in the visual and motor cortex, and possibly less in areas associated with social cognition and symbolic thought. though interpreting brain shape from fossil skulls is treacherous and conclusions are always tentative. They were skilled hunters, capable of taking down large and dangerous prey, working cooperatively in ways that required planning and communication and trust. They made sophisticated stone tools, controlled fire, built shelters, buried their dead sometimes with what appear to be grave goods, suggesting some form of ritual or belief about what comes after death. They cared for injured and elderly members of their groups, as evidenced by skeletons showing healed fractures and signs of long-term disability that could only have been survived with help from others. In short, they were people, recognizable as such even across the vast gulf of time and evolutionary divergence. And when modern humans encountered them, the encounters must have been complex, charged with all the possibilities and dangers that arise when two similar but distinct groups meet with overlapping needs and limited resources. Were they peaceful? Were they violent? Did they trade and cooperate, or did they compete and clash? The archaeological record is too sparse and too ambiguous to answer definitively, but the genetic record is clear on one point. They interbred, and they did so multiple times over thousands of years, leaving a permanent mark on the human genome. The first encounters probably happened in the Middle East, in the Levant, where the geographic corridors out of Africa narrow, and where both Neanderthals and modern humans left traces in the archaeological record during overlapping time periods. Fossil sites show evidence of both groups using similar tools, occupying the same caves at different times, living in landscapes that were lusher then than they are now, with more rivers and vegetation, supporting larger populations of both humans and the animals they hunted. At some point, in ways we can only imagine, individuals from the two groups met. Perhaps it was a chance encounter at a water source, a confrontation over territory that ended in negotiation rather than violence. Perhaps it was a raid, a violent taking of captives that led to forced unions. Perhaps it was something closer to trade or alliance, groups that lived near each other, developing relationships over generations, with occasional marriages or liaisons cementing bonds between communities. The details are lost, but the result is preserved in every non-African genome sequenced today. Clear evidence of gene flow from Neanderthals into the modern human lineage. Those hybrid children, born from Neanderthal and modern human parents, were the bridge between species, carrying chromosomes from both lineages and embodying in their own biology the collision of two evolutionary experiments that had been running independently for over 500,000 years. They must have looked intermediate, perhaps inheriting the heavy build and brow ridges from one parent and the lighter frame and higher forehead from the other, or carrying a mosaic of features that marked them as different from both parental groups. How they were treated by either community, whether they were accepted or marginalized or valued for their unique position, we cannot know, but genetically they were viable. They survived to adulthood, they reproduced, and their children in turn survived and reproduced, passing along fragments of Neanderthal DNA that would persist through all the generations to come, gradually mixing with the much larger modern human gene pool, spreading through populations as they expanded across Europe and Asia, thinning out through recombination but never disappearing entirely. If interbreeding had happened just once or twice, the genetic legacy would be trivial, a few rare chromosomal segments carried by a handful of descendants, likely to be lost to drift within a few generations. But the genetic evidence suggests multiple interbreeding events, possibly occurring over several thousand years as modern human populations expanded through territories where Neanderthals still lived. Each time a child was born from such a union, each time that child survived and had children of their own, a new injection of Neanderthal DNA entered the modern human gene pool. Over time, as modern humans spread and Neanderthals declined, eventually disappearing from the fossil record around 30,000 years ago, those fragments of Neanderthal ancestry were carried forward by the survivors, by the people who went on to colonize all of Eurasia and eventually the world beyond. And today, tens of thousands of years later, those fragments are present in billions of people, a genetic echo of encounters that happened in a world of ice and megafauna and endless wilderness that no longer exists. From an evolutionary perspective, the question is not whether interbreeding happened, the genetic evidence settles that beyond doubt, but why Neanderthal DNA persisted at all and why it persisted in such a non-random pattern. Most hybrid offspring from closely related species carry genetic variants that are slightly incompatible with one or both parental genomes, variants that cause subtle problems with development or fertility or health. And natural selection tends to weed those variants out over time, leaving only the truly neutral or beneficial ones. If you randomly scattered Neanderthal genes across the modern human genome and let natural selection run for 2,000 generations, you would expect most of those genes to disappear, either because they were actively harmful or because they provided no advantage and were lost to random drift. And indeed, that is what happened to most Neanderthal DNA. The majority of the Neanderthal genome, the parts that coded for things that were simply different but not better or worse in the modern human context, has been gradually lost, diluted down, and mixed out until it represents only a small percentage of any individual's total genetic material. But the immune system genes stayed. They not only stayed, they increased in frequency, spread through populations, reached levels where they are now present in the majority of people in some regions. And that is not random. That is natural selection acting powerfully and consistently over thousands of years, and it tells us something crucial about the value of those particular Neanderthal genes. When modern humans moved into Eurasia, they encountered a new world of pathogens, microbes that had co-evolved with Neanderthals for 300,000 years, that had been in an evolutionary arms race with Neanderthal immune systems, adapting to evade Neanderthal defenses, while Neanderthal immune systems adapted to recognize and neutralize them. Modern humans arriving from Africa had no such history. Their immune systems had been shaped by African pathogens, by the bacteria and viruses and parasites of tropical and subtropical environments, by the specific microbial ecology of the savannas and forests where they evolved. They were walking into a disease environment they were not prepared for, and the cost was high. Imagine a small band of modern humans, 50 or 100 individuals, moving through what is now Turkey or the Balkans, following game, looking for caves to shelter in as winter approaches. They are skilled, they have fire and tools and cooperation, but they are not immune to the microbes that float in the air, that live in the soil, that infect the animals they hunt and butcher. They fall sick with respiratory infections, with fevers that spread through the group, with illnesses they have no names for and no remedies against. Some recover, many do not. Children die. Adults die. The group shrinks, loses knowledge, loses members who were carrying crucial skills, and the expansion stalls or reverses as survivors retreat to safer ground. Now imagine a similar band, but this one includes a few individuals with Neanderthal ancestry, hybrid descendants from earlier encounters. They carry immune system genes that Neanderthals had already refined through millennia of exposure to these same Eurasian pathogens, genes that recognize bacterial surface proteins common in European soils, genes that produce antiviral molecules effective against the specific strains circulating in northern climates, genes that regulate inflammatory responses in ways that fight infection without causing the kind of overwhelming immune reaction that kills as surely as the pathogen itself. This second group gets sick too, but fewer of them die. More of the children survive. More adults recover and can continue hunting, gathering, protecting the group. The difference is small in any single epidemic, but over generations it accumulates into a profound advantage. Natural selection does not care about purity or species boundaries. It cares only about what works, about what increases the odds of survival and reproduction, and in the environment that modern humans were trying to colonize, having Neanderthal immune genes worked. Individuals who carried them were more likely to survive infections, more likely to raise children who also survived, more likely to pass those genes forward to the next generation, and the next, until what had started as a rare hybrid quirk became a common feature of the population. The genes spread not because anyone understood their function or made deliberate choices, but through the simple, brutal arithmetic of differential survival. And today, when you get sick and your immune system responds, part of what saves you might be molecular machinery that your ancestors borrowed from another species, a species that is extinct in every visible way, but that still lives, distributed and invisible, in the code that builds and maintains your body. Those hybrid children carried something their pure Homo sapiens relatives did not, something that had been tested and refined in Eurasia for hundreds of thousands of years, something invisible at the time, but written into every cell of their bodies. Immune system genes that already knew how to fight the diseases of Europe and Asia because they had been doing exactly that, generation after generation, through the deep evolutionary time that separated Neanderthals from the African migrants. To understand why this mattered so profoundly, why these particular genes persisted when most other Neanderthal DNA was gradually lost, you need to understand what the immune system actually is and why it evolves faster, changes more dramatically, and varies more widely between populations than almost any other system in the body. Your immune system is not a single organ, but a distributed network of cells and molecules and signaling pathways that collectively distinguish self from non-self, that recognize the molecular signatures of bacteria and viruses and parasites, that coordinate attacks against invaders while trying desperately not to attack your own tissues, and that must constantly adapt because the pathogens are adapting too, evolving ways to hide from detection, to disable defenses, to exploit the very mechanisms meant to destroy them. This creates an evolutionary arms race that never ends, where every generation sees new variants in immune genes being tested against new variants in pathogen genes, where what worked last year might fail this year because the bacteria have changed their surface proteins or the virus has mutated its recognition sequences, where populations in different parts of the world face different microbial threats and evolve different solutions, creating immune system diversity that maps onto geography and history and the specific diseases that threatened survival in particular places. Your immune genes are among the most variable regions of your genome, the places where you are most different from your neighbors and your ancestors, precisely because they are under constant pressure to change, to stay one step ahead of the microbes that want to use your body as a home. And when two human populations have been separated for 600,000 years, living on different continents, facing completely different sets of pathogens, their immune systems will have diverged accordingly, each lineage accumulating mutations that help with the specific threats they face, each tuning their defenses to the particular microbial ecology of their environment. The Neanderthals had lived in Europe and Asia for at least 300,000 years by the time modern humans arrived, possibly longer, depending on how you define when the Neanderthal lineage truly became distinct from its ancestors. 300,000 years is 12,000 generations, 12,000 rounds of natural selection testing every immune variant against the pathogens circulating in Ice Age Eurasia, 12,000 opportunities for beneficial mutations to spread and harmful ones to be eliminated. The diseases they faced were shaped by climate, by the animals they hunted and lived near, by population density, by the specific bacteria that thrive in cold soils and the viruses that circulate in seasonal wildlife populations. They would have encountered respiratory infections that spread through groups huddled in caves during long winters, skin infections from wounds sustained in hunting dangerous megafauna, parasitic worms picked up from undercooked meat, zoonotic diseases jumping from the mammoths and cave bears and reindeer that shared their landscape, and countless other microbial challenges that we can only infer from modern disease ecology and from the genetic signatures those diseases left in Neanderthal immune systems. Through all those generations, through all those infections and epidemics and individual cases of sickness and recovery or death, natural selection was sculpting Neanderthal immune genes, favoring variants that recognized the most common bacterial surface molecules, that produced the right antiviral proteins at the right times, that balanced the inflammatory response to fight infection without causing so much inflammation that it killed the host. The genes that code for HLA molecules, the proteins that sit on cell surfaces and display fragments of whatever is inside the cell so that immune cells can scan for signs of infection, were under particularly intense selection. HLA genes are the most variable region of the human genome, with hundreds of different variants in modern populations because different variants recognize different pathogen molecules, and having diverse HLA types in a population means that at least some individuals will be able to recognize and fight any new pathogen that appears. Neanderthal HLA genes had been tuned by 300,000 years of Eurasian pathogens, shaped to recognize the specific proteins displayed by the bacteria and viruses most commonly encountered in their environment. The same was true for toll-like receptors, the TLR genes that sit on immune cell surfaces and in internal compartments, constantly sampling the molecular environment, looking for the characteristic patterns that mark invaders. TLR1 recognizes lipoproteins from bacterial cell walls. TLR6 and TLR10 recognize similar molecules from different bacterial species. TLR7 and TLR8 detect viral RNA. Each receptor is fine-tuned to bind specific molecular patterns, and small changes in the receptor's shape can alter what it recognizes and how strongly it responds. Neanderthal TLR genes carried variants that modern humans from Africa did not have, variants that almost certainly reflected adaptation to the specific pathogens present in Eurasia, though we can only speculate about exactly which diseases drove which changes because the microbes themselves left no direct fossil record, only their fingerprints in the genes of the hosts they infected. Then there are the OAS genes, the oligoadenylate synthetases that form part of the antiviral defense system, activated when viral RNA is detected inside cells, producing molecules that in turn activate enzymes that chop up the viral genetic material and shut down protein synthesis to starve the virus. The OAS gene family sits in a cluster on chromosome 3, and this cluster is one of the clearest examples of functional Neanderthal DNA persisting at high frequency in modern populations. Certain combinations of variants at this cluster, certain haplotypes that represent stretches of chromosome inherited as a unit, are unmistakably Neanderthal in origin, more similar to the sequences found in ancient Neanderthal bones than to the sequences found in African populations, and these Neanderthal haplotypes are common in Europe and Asia, present in 30 to 40% of people in some regions, which means they must be doing something useful, must provide enough benefit that natural selection has maintained them for 50,000 years, despite the general trend of Neanderthal DNA being diluted and discarded. When anatomically modern humans walked into this environment, their immune systems were calibrated for Africa, for the pathogens of warm climates, for the specific disease ecology of the tropics and subtropics, where they had spent the previous 200,000 years. They carried HLA variants that recognized African pathogens, TLR variants tuned to the bacteria and viruses common in equatorial regions, OAS variants optimized for the RNA viruses that circulated in dense populations living near rainforests and savannas. These were not bad immune systems, not weak or deficient, but they were specialized, adapted to one environment, and that environment was very different from the one they were entering. The bacteria in European soils had different surface proteins. The viruses circulating in northern animal populations had different RNA structures. The parasites that infected cold-adapted prey species had different life cycles. Modern human immune systems, encountering these pathogens for the first time, would have responded, but slowly, inefficiently, learning on the fly, losing people in the process of figuring out how to recognize and fight threats they had never seen before. This is where the hybrid advantage became critical, where the Neanderthal immune genes carried by some individuals made the difference between a population that struggled and declined and one that survived and expanded. Consider an early modern human group trying to establish itself in what is now southern France, 45,000 years ago, during a period when the climate was fluctuating and Neanderthals were still present in the region, though declining in numbers. Winter comes, the group retreats to caves, and someone gets sick with a respiratory infection, a virus or bacterium that has been circulating in European animal populations for millennia, and that Neanderthals have evolved defenses against. In most of the group, individuals with purely modern human immune genes, the pathogen spreads rapidly because their TLR receptors don't recognize it as efficiently. Their initial immune response is slower, and by the time the adaptive immune system figures out how to make antibodies, the infection has caused serious damage. But in a few individuals, those with Neanderthal ancestry, who carry Neanderthal TLR variants, the receptors recognize the pathogen immediately, the innate immune response kicks in within hours, antiviral proteins are produced, and the infection is contained before it becomes severe. The difference might be subtle in any single infection, but over years and decades, as the group encounters pathogen after pathogen, as children are exposed to diseases that kill some and spare others, as adults fight infections while also trying to hunt and gather and maintain the group, the cumulative effect is enormous. Families with Neanderthal immune variants have higher child survival rates. Adults in those families are sick less often, contribute more to the group's food supply, are better able to care for dependents. More of their children survive to reproductive age. Those children inherit the protective Neanderthal variants and pass them to their own children. Within a few dozen generations, the frequency of Neanderthal immune variants in the population has climbed from perhaps 5 or 10%, the initial level from limited interbreeding, to 30 or 40 or 50%. Not because anyone chose to favor Neanderthal genes, but because natural selection, indifferent and mechanical, simply amplified whatever worked, and in the disease environment of Ice Age Eurasia, those Neanderthal immune genes worked better than the modern human alternatives. This is adaptive introgression operating at maximum efficiency, a textbook example of how hybrid vigor can arise when two populations that have adapted to different environments interbreed and their offspring inherit the best adaptations from both sides. The modern humans brought advantages of their own, possibly including more sophisticated language, more complex social structures, better long-range hunting weapons, and those advantages helped them spread and eventually dominate. But they also brought vulnerabilities, gaps in their adaptive toolkit, where the Neanderthals, for all their eventual extinction, had solutions that modern humans lacked. The immune system was the clearest and most consequential of those gaps, and filling it with borrowed Neanderthal genes might have made the difference between successful colonization of Eurasia and repeated failed attempts that ended in disease-driven population crashes. The Neanderthals, in a very real sense, gave modern humans the biological passport they needed to survive in environments that would otherwise have killed them, and the proof is written in every genome, in the elevated frequencies of Neanderthal immune variants that natural selection preserved and amplified while discarding most of the rest of the Neanderthal legacy as incompatible or unnecessary. But not all Neanderthal DNA was beneficial, and in fact, most of it was quietly discarded by natural selection over the 50,000 years since interbreeding first occurred, scrubbed out of modern human genomes through a slow but relentless filtering process that reveals something important about hybrid compatibility and the limits of mixing between long-separated lineages. If you could rewind time to the moment when the first hybrid children were born, when their cells divided and their chromosomes sorted into eggs and sperm that would create the next generation, you would see a genome that was perhaps 10 or 20% Neanderthal, large blocks of chromosomes inherited intact from a Neanderthal parent sitting alongside modern human chromosomes from the other parent. That was the starting point, the raw material that natural selection would work with over the next 2,000 generations. Today, when we sequence genomes from people of European or Asian descent, we find only 1 to 4% Neanderthal DNA, and that remaining fraction is not randomly distributed, but concentrated in specific regions, creating a landscape of peaks and valleys that tells the story of what was useful enough to keep and what was incompatible enough to discard. The valleys, the regions where Neanderthal DNA is almost completely absent, cluster near genes that are essential for basic biological functions, particularly reproduction and development. Near genes involved in sperm production and testis function, Neanderthal ancestry drops to nearly zero, suggesting that Neanderthal variants in these regions caused fertility problems in hybrid males, reducing their ability to have children and thus automatically eliminating those variants from the gene pool within a few generations. The same pattern appears near genes involved in female reproduction, in placental function, in the complex molecular choreography that must unfold correctly for a pregnancy to succeed and a healthy baby to be born. Hybrid incompatibility in reproductive genes is one of the first barriers that arise when populations diverge, a sign that 600,000 years of separate evolution had taken Neanderthals and modern humans to the edge of being able to produce fully viable offspring. Most hybrids were probably healthy and fertile, but some fraction carried combinations of Neanderthal and modern human genes that did not work well together, and those individuals left fewer descendants, and their problematic gene combinations were gradually removed from the population. Brain development genes show a similar pattern of Neanderthal depletion, though the reasons are more speculative and harder to interpret. Neanderthal brains were large, possibly larger on average than modern human brains in raw volume, but they were shaped differently, with more development in visual and motor processing regions, and possibly less in areas associated with social cognition and symbolic thought, though inferring cognitive differences from skull shape is fraught with uncertainty. Whatever the functional differences, the genetic variants that control brain growth and neural development in Neanderthals were apparently not fully compatible with the modern human developmental program. Perhaps they caused subtle problems with timing, with the coordination of growth across different brain regions, with the formation of neural connections that modern human cognition depends on. Perhaps the incompatibility was social rather than strictly biological, if Neanderthal cognitive variants produced behaviors or abilities that were disadvantageous in modern human societies that were becoming more complex, more language-dependent, more reliant on large-scale cooperation. We do not know, and we may never know with certainty, but the genetic evidence is clear. Neanderthal DNA near brain development genes was selected against, removed from the population, present today at frequencies far below what random chance would predict. Across much of the rest of the genome, in regions coding for basic metabolic functions, structural proteins, housekeeping genes that every cell needs to function, Neanderthal DNA simply drifted, neither strongly favored nor strongly disfavored, gradually diluted through recombination and mixing until it reached the low background levels we see today. These were the neutral regions, places where Neanderthal variants worked fine, but provided no particular advantage over the modern human equivalents, and without positive selection to maintain them, they slowly faded, replaced generation by generation with modern human sequences simply because modern humans vastly outnumbered the small initial hybrid population, and in any random mating, the odds favored inheriting the more common modern human version of any given gene. The result, after 50,000 years, is a genome where the majority of Neanderthal ancestry has been erased, leaving only scattered fragments that are individually tiny, but collectively still add up to millions of base pairs, enough to affect hundreds or thousands of traits, enough to make a measurable difference in biology and health. But the immune system is the glaring exception, the place where natural selection not only tolerated Neanderthal DNA, but actively favored it, drove it to higher and higher frequencies, preserved it across the millennia while discarding most everything else. When researchers map Neanderthal ancestry across modern genomes and look for regions where it is unusually common, where it appears in far more people than chance would allow, the peaks cluster overwhelmingly in immune system genes. The HLA region on chromosome 6, which codes for the major histocompatibility complex proteins that display pathogen fragments on cell surfaces, shows some of the highest Neanderthal ancestry in the entire genome. Some HLA variants found in Europeans and Asians are nearly identical to variants found in Neanderthal fossils, and are clearly the result of introgression rather than independent mutation, and these introgressed variants are present in 30, 40, sometimes over 50% of individuals in certain populations, frequencies that can only be explained by strong positive selection maintaining them over thousands of generations. The functional logic is straightforward once you understand what HLA molecules do. They are the presentation system for the adaptive immune system, the mechanism by which cells display samples of their internal contents on their surfaces so that T cells can inspect them and determine whether the cell is healthy or infected. Each HLA variant has a slightly different binding groove, a pocket that fits certain peptide shapes and not others, and that specificity determines which pathogen fragments can be displayed, and thus which infections the immune system can recognize and respond to quickly. Having diverse HLA types in a population is advantageous because it means that at least some individuals will be able to display peptides from any new pathogen that emerges, and those individuals will mount an immune response while others might not. Neanderthal AHN HLA variants represented additional diversity, new shapes for the binding grooves, coverage for pathogen peptides that modern human HLA variants might miss, and in a disease environment where modern humans were encountering Eurasian pathogens for the first time, that extra coverage was life-saving. The same pattern appears in the TLR gene family, the toll-like receptors that form the first line of innate immune defense, the sensors that detect bacterial and viral molecules and trigger immediate inflammatory responses before the slower adaptive immune system has time to mobilize. TLR1, TLR6, and TLR10 sit in a cluster on chromosome 4, and Neanderthal variants in this cluster are common in modern Europeans and Asians, present in substantial fractions of the population, clearly maintained by selection. These receptors recognize lipoproteins and other molecules on bacterial surfaces, and the Neanderthal variants apparently provided better or faster recognition of the bacteria present in Eurasian environments, triggering immune responses that helped clear infections before they became severe. Small differences in receptor shape can translate into large differences in how quickly and strongly the immune system responds to a pathogen, and in marginal survival conditions, that speed matters enormously. Then there is the OAS gene cluster on chromosome 12, a family of antiviral genes that produce enzymes activated by viral RNA, enzymes that in turn trigger the destruction of viral genetic material and shut down cellular protein synthesis to starve the virus. Neanderthal haplotypes at this locus, long stretches of linked variants inherited as a unit, are present at frequencies well above neutral expectations in European and Asian populations, another clear signature of positive selection. The OAS genes are part of the interferon response, the antiviral defense system that ramps up when cells detect foreign RNA, and having Neanderthal variants that were pre-adapted to Eurasian viruses would have provided immediate protection against infections that killed modern humans who lacked those variants. Even today, 50,000 years later, those Neanderthal OAS variants affect how strongly people respond to viral infections, how much interferon their cells produce, how quickly they clear viruses from their systems, and those effects have real consequences for health and disease in the modern world, as we will see when we examine specific cases like COVID-19. The cumulative picture from population genetics is unambiguous. Most Neanderthal DNA was neutral or harmful in a modern human context and was gradually eliminated by selection or drift, creating deserts near reproductive and developmental genes and thinning to low levels across most of the genome. But immune system genes were the exception, the place where Neanderthal ancestry was not just tolerated but actively amplified, where natural selection saw clear value and acted to preserve and spread those variants through populations. The modern human immune system in people of non-African descent is, in a very real sense, a hybrid system, built partly from genes that evolved in Africa over hundreds of thousands of years, and partly from genes borrowed from Neanderthals, who had spent an equally long time adapting to Eurasian diseases. It is not purely Homo sapiens and never was, and that mixture, that borrowed diversity, might have made the difference between successful colonization of the northern hemisphere and repeated failures, between survival and extinction for the modern human migrants who gambled everything on expanding beyond their ancestral homelands. The clearest example of this ancient genetic legacy shaping modern health is a cluster of immune genes on chromosome 3, a Neanderthal gift that still determines who gets desperately sick and who barely notices when certain viruses sweep through populations, a stretch of DNA that remained invisible and unremarked for 50,000 years until a novel coronavirus forced scientists to ask why some people ended up on ventilators while their neighbors with identical exposure and similar health backgrounds walked away with mild symptoms or none at all. The OAS gene family, clustered together on this chromosome, codes for enzymes with a specific and crucial job: detecting viral RNA inside cells and triggering a cascade of antiviral responses that can stop an infection before it overwhelms the body. These are ancient genes present in all mammals, part of the interferon system that represents one of evolution's oldest solutions to the problem of viruses, but the specific variants that many Europeans and Asians carry, the particular versions of these genes that determine how strongly and how quickly they activate, are not purely modern human in origin. They are Neanderthal, borrowed from a species that spent hundreds of thousands of years fighting Eurasian viruses. And they are present at high frequencies today because they worked, because they saved lives, because natural selection recognized their value and amplified them through populations even as it was discarding most other Neanderthal DNA as incompatible or useless. When a virus enters your cells, when its genetic material starts hijacking your cellular machinery to produce more copies of itself, one of the first alarm systems to detect the intrusion is a family of proteins that recognize viral RNA, the genetic blueprints that many viruses carry instead of DNA, molecules that have characteristic shapes and chemical modifications that mark them as foreign. The OAS proteins are part of this detection system, activated when they bind to viral RNA, and once activated, they produce small signaling molecules that in turn activate an enzyme called RNase-L, which starts chopping up all the RNA in the cell, both viral and cellular, essentially scorching the earth to deny the virus the resources it needs to replicate. At the same time, the interferon signaling cascade ramps up, warning neighboring cells to prepare their defenses, recruiting immune cells to the site of infection, creating an antiviral state that makes it harder for the virus to spread through tissues. The speed and strength of this response matter enormously. The difference between a virus being contained in the initial cluster of infected cells and spreading systemically through the body, and that speed and strength are determined in part by which variants of the OAS genes you carry. The Neanderthal haplotype at this locus, the specific combination of variants linked together across this chromosomal region, produces OAS enzymes that are slightly different in structure and function from the modern human versions, and those differences translate into measurably different antiviral responses. People who carry the Neanderthal haplotype tend to produce more OAS protein when infected with RNA viruses, tend to activate the RNase-L pathway more strongly, tend to clear viral infections faster and with less viral load than people who carry only modern human variants. This is not a subtle effect, not a statistical whisper that only shows up in massive population studies, but a substantial functional difference that can be measured in cell culture experiments, in studies of immune response markers, in clinical outcomes during viral epidemics. The Neanderthal variants provide better antiviral defense, at least against certain types of RNA viruses, and that advantage has been maintained by natural selection for 2,000 generations because viruses have been killing humans for all of recorded history and far beyond, and anything that improves survival during viral pandemics will be favored by evolution, will spread through populations, will persist even when most other genetic material from the same source is being discarded. This remained an interesting but abstract finding, a footnote in evolutionary genetics papers until the winter of 2019 and 2020, when a novel coronavirus emerged in Wuhan, China, and spread globally within months, forcing the entire world into lockdowns and quarantines and desperate searches for treatments and vaccines. And scientists began asking why COVID-19 severity varied so wildly between individuals. Age mattered, underlying health conditions mattered, but even accounting for those factors, the variation was enormous. Some young, healthy people died. Some elderly people with multiple health problems had mild cases. The search for genetic factors that might explain this variation led researchers to genome-wide association studies, massive efforts to compare the genomes of people with severe disease to those with mild or asymptomatic infections, looking for any genetic variants that appeared more often in one group than the other, and one of the strongest signals that emerged pointed directly at chromosome 3, at the OAS gene cluster, at the exact region where Neanderthal ancestry is common in Eurasian populations. The pattern that emerged was complex, revealing that Neanderthal immune genes are not simple blessings or curses, but context-dependent tools whose value depends on the specific pathogen and the specific circumstances of infection. The Neanderthal haplotype on chromosome 3 The OAS cluster that confers stronger antiviral responses turned out to be protective against severe COVID-19, associated with lower rates of hospitalization, less need for intensive care, better survival outcomes. People who inherited this Neanderthal variant, which is present in about 30% of people in East Asia and perhaps 10% in Europe, were measurably less likely to develop life-threatening complications from coronavirus infection, all else being equal. Their cells, armed with Neanderthal OAS genes, detected the viral RNA faster, produced more interferon, activated antiviral pathways more strongly, and cleared the virus more efficiently, limiting the damage before it could progress to the devastating lung inflammation and systemic complications that killed millions worldwide. But the Neanderthal legacy is not uniformly protective, not a simple gift of better immunity, because on a different chromosome, in a different cluster of immune genes, other Neanderthal variants had the opposite effect, increasing rather than decreasing the risk of severe COVID-19. On chromosome 12, variants in genes that regulate inflammatory responses, that control how strongly the immune system reacts to infection, showed clear Neanderthal ancestry, and these variants were associated with higher rates of severe disease, with the dangerous cytokine storms where the immune system overreacts, producing so much inflammation that it damages the lungs and other organs as badly as the virus itself. These Neanderthal variants, which might have been beneficial in fighting bacterial infections or other pathogens where strong inflammation is protective, became liabilities in the context of COVID-19, where excessive immune activation is often more dangerous than the virus, and people who carried them were more likely to end up in intensive care, more likely to need ventilators, more likely to die. The image that emerges is of a genome that is fundamentally hybrid, carrying immune variants from multiple sources, some helpful and some harmful depending on the specific threat. And the outcome of any particular infection depends on which combination of variants you happen to have inherited from the complex lottery of sexual reproduction and ancestral mixing. Two people, genetically similar in most respects, both healthy, both the same age, might encounter the same virus and have radically different outcomes, not because of chance or environmental factors, but because one carries Neanderthal chromosome 3, OAS variants that help clear the virus quickly, while the other carries Neanderthal chromosome 12 inflammatory variants that cause dangerous overreaction, or because one has the protective variants and not the harmful ones, while the other has the reverse. The differences are measurable, clinically significant, and entirely invisible unless you sequence the genome and look for those specific Neanderthal markers, those fragments of extinct DNA still shaping life and death in the 21st century. This realization has transformed how scientists think about human genetic diversity and disease susceptibility, moving away from the old assumption that there is one normal human genome with minor variations and toward an understanding that human populations are mosaics, carrying different combinations of ancestral variants shaped by different histories, different diseases, different selective pressures, and that those differences matter medically. A drug or treatment that works well in one population might work poorly in another because they have different immune gene variants. A vaccine that produces strong antibody responses in people with one genetic background might produce weaker responses in people with another. Risk prediction models that were built using European populations who have certain Neanderthal variants at certain frequencies will not work as well in Asian populations where the same variants appear at different frequencies or where different Neanderthal variants are present. Precision medicine, the effort to tailor treatments to individual genetics, must account for this Neanderthal legacy, must recognize that extinct humans are still influencing clinical outcomes, still determining who responds to interferon therapy, who suffers severe reactions to certain infections, who benefits from aggressive versus moderate immune suppression. The COVID-19 pandemic became, in a strange and unintended way, the largest natural experiment ever conducted on the functional consequences of carrying Neanderthal DNA, an involuntary test of which ancient variants still provide value and which have become liabilities in the modern disease environment. The results have been sobering, revealing that adaptive introgression 50,000 years ago shaped who survived and who didn't in 2020, that evolution is not just ancient history, but an ongoing process whose effects ripple forward through time, connecting us to extinct relatives in ways we are only beginning to understand. Every person who survived severe COVID-19 because they carried protective Neanderthal variants, every person who died because they carried risk-increasing variants, is evidence that the Neanderthals are not gone, not fully extinct, but living distributed through billions of bodies, still fighting pathogens, still shaping outcomes, still mattering in the most fundamental ways possible. Life, death, survival, extinction. And the story is not finished, because new viruses will emerge, new pandemics will test human populations, and each time, part of who survives will be determined by these ancient genes, these borrowed sequences, these ghosts still breathing through us. But the Neanderthal immune legacy is not always a gift, and sometimes in the clean, sanitized environments of the modern developed world, what once saved lives becomes the source of chronic suffering, a double-edged sword that protects against some threats while creating others that our ancestors could never have anticipated. The same hyperactive immune responses that helped Neanderthals and their hybrid descendants survive bacterial infections in Ice Age caves, the same hair trigger inflammation that cleared pathogens quickly before they could spread through vulnerable populations huddled together through long winters, can turn inward in a world where most infectious diseases have been controlled, where children grow up in homes cleaned with antimicrobial products, where antibiotics eliminate bacterial threats before the immune system needs to fight them, and the result is an immune system that has been tuned by evolution to fight enemies that no longer appear with the frequency they once did, leaving it restless, overreactive, prone to attacking harmless substances or even the body's own tissues in a desperate search for something to do. This is the autoimmune paradox, and it is written partly in Neanderthal DNA, in those same TLR genes and inflammatory response pathways that provided such powerful advantages against infections, but that become liabilities when the microbial landscape changes. Autoimmune diseases, conditions where the immune system mistakenly attacks the body's own cells and tissues, have risen dramatically in developed countries over the past century, and while environmental factors like diet, pollution, antibiotic use, and reduced microbial exposure certainly play major roles, genetics matters too. And when researchers conduct genome-wide association studies looking for genetic variants that increase autoimmune disease risk, they keep finding Neanderthal DNA. The same variants that helped clear infections by producing strong inflammatory responses turn out to increase the risk of Crohn's disease, an inflammatory bowel disorder where the immune system attacks the intestinal lining. The same variants that ramped up immune activation against pathogens increase susceptibility to lupus, where antibodies attack the body's own DNA and proteins. The same hyperreactive immune genes that protected against bacterial invasion raise the odds of type 1 diabetes, where immune cells destroy the insulin-producing cells of the pancreas. The mechanism is straightforward once you understand what these genes do and how their context has changed. Toll-like receptors, those pattern recognition molecules that detect bacterial and viral signatures and trigger immediate inflammatory responses, come in different sensitivities depending on which genetic variants you carry. The Neanderthal variants tend to be more sensitive, requiring less stimulation to activate, producing stronger signals when they do detect something, recruiting more immune cells more quickly to sites of potential infection. In a world teeming with dangerous pathogens, where a scratch from a mammoth tusk could introduce flesh-eating bacteria, where contaminated water carried dysentery, where close living quarters spread respiratory infections that could kill half a group, having sensitive hair-trigger immune responses was protective, the difference between containing an infection early and dying from sepsis or pneumonia. Natural selection strongly favored those variants, driving them to high frequencies in populations that faced constant microbial threats. But the modern developed world is not teeming with dangerous pathogens, or at least not to the same degree. Water is treated and safe. Food is refrigerated and inspected. Wounds are cleaned with antiseptics and covered with sterile bandages. Antibiotics eliminate most bacterial infections before they become life-threatening. Children are vaccinated against diseases that once killed millions. The average person in a wealthy country encounters far fewer serious pathogens in a lifetime than a Neanderthal child would have faced in a single year. And in this low-pathogen environment, an immune system calibrated for constant warfare has nothing appropriate to fight. The Neanderthal TLR variants still sit on cell surfaces, still scan constantly for bacterial lipoproteins and viral RNA, still trigger inflammation when they detect molecular patterns. But now the patterns they detect are often harmless. Proteins in pollen, molecules in certain foods, even the normal bacteria that live peacefully in the gut without causing harm. The immune system responds as if these harmless substances were deadly threats, launching attacks that damage tissues and create chronic inflammation that persists for years or decades. Allergies are the mildest manifestation, immune overreactions to harmless environmental proteins that result in runny noses, itchy eyes, skin rashes, and in severe cases, life-threatening anaphylaxis, where airways swell shut and blood pressure drops. The prevalence of allergies has skyrocketed in developed countries, affecting up to 30 or 40% of children in some regions. And while environmental factors like pollution and diet contribute, genetic susceptibility plays a major role, and Neanderthal immune variants appear repeatedly in studies of allergy risk. Asthma, where airways become inflamed and constrict in response to allergens or irritants, shows similar patterns, with certain Neanderthal variants associated with increased risk of developing chronic asthma, particularly the severe forms that require daily medication and can cause life-threatening attacks. Eczema, an inflammatory skin condition that causes itching, redness, and cracked skin, also links to Neanderthal immune genes, creating misery for millions of children and adults whose skin overreacts to minor irritants or even to their own skin bacteria. The more severe autoimmune diseases tell an even starker story of adaptation turned maladaptive. Crohn's disease and ulcerative colitis, collectively called inflammatory bowel disease, involve chronic inflammation of the digestive tract, where immune cells attack the intestinal lining as if it were infected, creating ulcers, bleeding, severe pain, and in many cases requiring surgery to remove damaged sections of bowel. These diseases are far more common in populations of European descent than in African or Asian populations, and genetic studies have identified multiple Neanderthal variants that increase risk, particularly in genes that regulate how strongly the immune system responds to gut bacteria. In the ancestral environment, where food was often contaminated and intestinal infections common, having a strong immune response in the gut was protective, clearing pathogens before they could cause systemic illness. In the modern sterile environment, where food is clean and gut bacteria are mostly beneficial, that same strong response becomes destructive, treating normal bacteria as invaders and creating inflammation that never resolves. Lupus, an autoimmune disease where the immune system produces antibodies against the body's own DNA, proteins, and cell membranes, causing inflammation in joints, skin, kidneys, and other organs, shows elevated rates in people with certain Neanderthal immune variants, particularly those affecting interferon production and inflammatory signaling. Type 1 diabetes, where immune cells destroy the insulin-producing beta cells in the pancreas, leaving patients dependent on insulin injections for life, has genetic risk factors that include Neanderthal variants in immune regulation genes. Multiple sclerosis, where the immune system attacks the protective myelin sheaths around nerve fibers in the brain and spinal cord, causing progressive neurological damage, shows associations with Neanderthal variants in some populations. The pattern repeats across dozens of autoimmune and inflammatory conditions. Genes that were adaptive in one context become harmful in another, and the people who suffer are paying the price for evolutionary adaptations that no longer match their environment. Consider a young woman in Northern Europe, born with a genome that includes multiple Neanderthal immune variants inherited from both parents, variants that her ancestors carried successfully for thousands of years, variants that helped them survive plagues and infections that swept through medieval villages. She grows up in a clean, modern home, vaccinated against major diseases, eating safe food, drinking treated water. Her hyperactive immune system, primed by evolution to fight constant microbial threats, finds few real enemies. Instead, it begins attacking pollen in spring, causing severe hay fever that makes her eyes swell shut and her nose run constantly. By her 20s, she develops eczema that covers her hands and arms with itchy, painful rashes. In her 30s, she starts experiencing joint pain and fatigue, eventually diagnosed with lupus, her immune system producing antibodies that attack her own connective tissue. She requires immunosuppressive drugs to control the inflammation, medications that dampen her immune system and make her more vulnerable to the very infections her Neanderthal genes evolved to fight. A cruel irony where the treatment for having too much immunity is to suppress it artificially, recreating a less reactive state that would have been normal in a human without those particular Neanderthal variants. Now compare her to someone of recent African ancestry whose genome carries no Neanderthal DNA because their ancestors never left Africa and never encountered Neanderthals, whose immune genes followed a different evolutionary path shaped by African diseases, particularly by malaria and other tropical pathogens. This person might have different immune challenges, different disease susceptibilities shaped by their own ancestral environment, but on average, they have lower rates of the autoimmune diseases that plague European populations, lower rates of severe allergies and inflammatory bowel disease, because they lack the specific Neanderthal variants that predispose to hyperactive immune responses. This is not about one genetic background being superior to another, but about different populations carrying different adaptive legacies that create different vulnerabilities in the modern environment, and understanding those differences matters for medicine, for treatment, for predicting who is at risk and why. The tragedy is that what were once life-saving adaptations, gene variants that increased survival and allowed human expansion into new continents, have become sources of chronic disease in a world that changed faster than evolution could keep up. Neanderthal immune genes did exactly what evolution designed them to do. They fought infections, they saved lives, they spread through populations because they worked. But evolution is blind, cannot anticipate future environments, cannot know that 50,000 years later, the disease landscape would be completely different, that the threats would shift from too many pathogens to too few, from deadly infections to sterile inflammation, from fighting enemies outside the body to the body attacking itself. The Neanderthals who survived long enough to pass these genes forward could not have imagined a world where their great-great-grandchildren, 1,000 generations removed, would suffer because the immune system they bequeathed was too strong, too sensitive, too ready to fight. And the modern descendants carrying those genes cannot simply evolution them away, cannot wait for natural selection to adjust because autoimmune diseases mostly strike after reproductive age, or at least don't reduce reproduction enough to create strong selection against them. We are stuck with this legacy, this borrowed DNA that was once salvation and has become, for some, a curse, and the only solutions are technological: drugs to suppress inflammation, treatments to calm overactive immune systems, and perhaps someday, gene therapies to edit out the problematic variants while keeping the protective ones, to separate the gift from the curse that Neanderthal extinction left behind. The Neanderthal contribution wasn't limited to a few genes, and it didn't affect all populations in the same way, because the interbreeding events that introduced Neanderthal DNA into modern humans happened in different places, at different times, involving different Neanderthal populations that had themselves diverged and adapted to different regional environments across the vast expanse of Ice Age Eurasia. When researchers began mapping Neanderthal ancestry in detail across global populations, they discovered something unexpected. Not only do people of African descent carry essentially no Neanderthal DNA, while people of European and Asian descent carry 1 to 4%, but the specific Neanderthal variants present in East Asian populations are partially different from those in European populations, different enough that the immune systems of these two groups, both carrying Neanderthal genes, respond differently to the same pathogens, creating patterns of disease susceptibility that map onto geography and ancestry in ways that have profound implications for medicine, for drug development, for understanding why certain populations are hit harder by certain diseases, while others seem relatively protected. The explanation lies in the complex history of human migration and mixing. When modern humans first left Africa, probably around 60,000 years ago, They encountered Neanderthals somewhere in the Middle East, in the Levant, or perhaps further east, and some interbreeding occurred, introducing Neanderthal DNA into the ancestral population that would eventually give rise to all non-African peoples. But that was not the only encounter. As modern humans continued spreading, some groups moved west into Europe, encountering Neanderthal populations that had been living there for hundreds of thousands of years, adapting to the specific cold, forested environments of glacial Europe. Other groups moved east across Asia, encountering different Neanderthal populations that had adapted to the steppes, the river valleys, the varied climates of Central and Eastern Eurasia. These different Neanderthal populations, separated by thousands of miles and tens of thousands of years of independent evolution, carried somewhat different immune gene variants, shaped by the different pathogens they encountered in their respective regions. And when they interbred with modern humans, they introduced different adaptive variants into different human populations. The result is that Europeans and East Asians both carry Neanderthal immune genes, but not exactly the same ones. In the TLR gene cluster on chromosome 4, the specific variants that are common in Europe differ in some positions from the variants common in East Asia, reflecting introgression from different Neanderthal source populations. In the HLA region, the major histocompatibility complex that determines which pathogen fragments the immune system can recognize and display, the Neanderthal-derived alleles present in high frequencies in Asian populations include some that are rare or absent in Europeans, and vice versa. These are not trivial differences, not just genetic curiosities, but functional variations that affect how strongly and how quickly the immune system responds to specific bacteria and viruses, and those differences translate into measurably different health outcomes when populations encounter the same diseases. Hepatitis B, a viral infection that attacks the liver and can lead to chronic disease, cirrhosis, and liver cancer, provides a clear example of these ancestry-based differences in immune response. Chronic hepatitis B infection, where the virus persists in the body for years or decades because the immune system fails to clear it completely, is far more common in East Asian populations than in European populations. Part of this reflects different patterns of transmission, with mother-to-child infection more common in some regions. But genetics plays a role too, and specific HLA types that are common in East Asia and that have signatures of Neanderthal or Denisovan origin are associated with higher rates of chronic infection, suggesting that these particular immune variants, whatever advantages they provided against other pathogens in the ancestral environment, are less effective at clearing hepatitis B. Europeans, carrying different HLA variants from different Neanderthal source populations, have on average better ability to mount an immune response that clears the virus before it becomes chronic. HIV, the virus that causes AIDS, shows a different pattern. Certain HLA alleles found predominantly in European populations, some with clear Neanderthal ancestry, are associated with slower progression from HIV infection to full-blown AIDS, meaning that people who carry these variants can live longer without treatment after initial infection because their immune systems control the viral replication more effectively. These protective HLA types are much rarer in African populations, which lack Neanderthal ancestry, and in some Asian populations, which carry different Neanderthal HLA variants, creating disparities in disease progression that have nothing to do with behavior or access to care, but are written in the genome, inherited from extinct humans who never encountered HIV, but whose immune genes happened to provide cross-protection against a modern virus. The irony is profound. Neanderthals, extinct for 30,000 years, are still affecting who survives HIV in the 21st century, their genes providing unintended benefits against a pathogen that did not exist when they walked the earth. Tuberculosis, one of the oldest and deadliest bacterial infections affecting humans, kills differently depending on ancestry, and Neanderthal immune genes are part of the explanation. The inflammatory response to TB infection, the formation of granulomas in the lungs where immune cells wall off infected areas to prevent bacterial spread, varies in intensity and effectiveness between populations, and genetic studies have linked some of this variation to Neanderthal TLR variants that affect how strongly the immune system responds to bacterial molecules. Europeans, with certain Neanderthal inflammatory variants, tend to mount stronger, faster inflammatory responses that can contain the infection but also cause more lung damage. East Asians with different variants may respond more slowly but with less tissue destruction. Neither response is uniformly better. Each reflects a different evolutionary trade-off, a different balance between fighting the pathogen and limiting collateral damage. And understanding these differences matters for treatment, for predicting who will develop severe disease, for tailoring therapies to work with rather than against the immune responses that different genetic backgrounds predispose toward. Even susceptibility to common respiratory infections, the colds and flu viruses that sweep through populations every winter, shows ancestry-related patterns that trace partly to Neanderthal immune genes. The OAS gene cluster that protects against COVID-19 severe disease also affects responses to influenza and other RNA viruses, and the protective Neanderthal haplotype is present at about 30% frequency in East Asian populations, but only 10% in Europeans, potentially contributing to different patterns of flu severity between these groups, though environmental factors, previous exposure history, and other genetic differences also play major roles. The point is not that genetics determines everything, but that it contributes, often substantially, to the complex picture of why diseases hit different populations differently, and ignoring that genetic component because of discomfort with discussing biological differences between ancestry groups does not make the differences go away. It just makes medicine less effective, less precise, less able to predict and prevent bad outcomes. The reverse pattern appears for some autoimmune diseases, where Europeans show higher rates than East Asians, reflecting the different Neanderthal immune variants they carry. Crohn's disease, multiple sclerosis, lupus, all more common in people of European descent, all linked to specific Neanderthal variants that are either absent or present at different frequencies in Asian populations. This is not because Europeans are weaker or more prone to disease in some general sense, but because they carry specific immune gene variants that were adaptive in one context, fighting infections in Ice Age Europe, and have become maladaptive in another context, the modern low-pathogen environment where hyperactive inflammation causes more harm than good. East Asians, with different Neanderthal variants shaped by different ancestral environments, have different autoimmune risk profiles, higher rates of some conditions, and lower rates of others, reflecting their own unique evolutionary legacy. What emerges from all this genetic archaeology is a picture of human immune diversity that is far more complex than the old model of one human immune system with minor variations. Instead, we are a patchwork species, different populations carrying different combinations of modern human genes, Neanderthal genes, and in some cases Denisovan genes, all mixed together through tens of thousands of years of migration and interbreeding and selection, creating immune systems that are fundamentally different in their strengths and weaknesses, their sensitivities and blind spots, their trade-offs between fighting infections and avoiding autoimmunity. There is no single best immune system, no optimal configuration that would work equally well for everyone in every environment, against every pathogen. There are only different solutions to the same fundamental problem of survival in a world full of microbes, solutions shaped by different histories, preserved in different genomes, and still affecting who lives and who dies when diseases strike in the modern world. The Neanderthals are not equally present in all of us, not distributed uniformly across humanity, but concentrated in some populations and absent from others, and those differences matter medically, clinically, in ways we are only beginning to understand and account for in the practice of medicine. But Neanderthals were not the only ghost species that left immune genes behind, not the only extinct human lineage that contributed adaptive variants to modern populations through ancient interbreeding. And when scientists began looking more closely at the immune gene variants carried by people of Asian and Pacific ancestry, they found sequences that were clearly archaic, clearly not modern human in origin, but also not Neanderthal, not matching the variants found in European Neanderthal fossils or in the genomes of modern Europeans. These variants pointed to another source, another branch of the human family tree that had been completely unknown to science until 2010, when researchers extracted and sequenced DNA from a tiny finger bone found in Denisova Cave in Siberia and discovered that it belonged to a teenage girl who was neither Homo sapiens nor Neanderthal, but represented a sister lineage that had split from the ancestors of both groups roughly 600,000 years ago and had evolved independently ever since, spreading across Asia and leaving almost no fossil record but abundant genetic traces in the populations that eventually absorbed them through interbreeding. The Denisovans, named after the cave where that first bone was found, remain mysterious in ways that Neanderthals are not. Because while we have hundreds of Neanderthal skeletons and thousands of archaeological sites documenting their lives, we have only a handful of Denisovan bone fragments, a few teeth, a piece of skull, not enough to reconstruct what they looked like or how they lived in any detail. What we do have is their genome, painstakingly assembled from ancient DNA preserved in those few bones, and we have the fragments of that genome still present in living people, particularly in East Asia, Southeast Asia, Australia, and the Pacific Islands, where Denisovan ancestry reaches levels of 1 to 5%, higher than Neanderthal ancestry in those same populations, clear evidence that interbreeding between Denisovans and modern humans was substantial, and that natural selection preserved many of the introgressed variants because they provided survival advantages in the environments where those populations lived. The Denisovan contribution to the immune system follows the same pattern as the Neanderthal contribution, but involves different genes and different populations, reflecting the fact that Denisovans inhabited different parts of Asia than Neanderthals, adapted to different climates and pathogens, and passed different adaptive variants to the modern humans who encountered them. In the HLA region, that critical immune gene cluster on chromosome 6, some of the alleles present at high frequencies in East Asian, Southeast Asian, and Pacific populations are unmistakably Denisovan in origin, more similar to the sequences recovered from Denisovan fossils than to any modern human variants, and these Denisovan HLA alleles provide coverage for pathogen peptides that modern human HLA variants miss, expanding the range of infections that the immune system can recognize and respond to effectively. In some Papua New Guinea populations, where Denisovan ancestry is highest, reaching 4 or 5% of the genome, Denisovan HLA alleles are present in more than half the population, maintained at these extraordinary frequencies by natural selection because they provide protection against local diseases. One specific example that has been studied in detail involves TNF-AIP3, a gene that regulates inflammatory responses and prevents the immune system from overreacting, a molecular break that limits the damage from excessive inflammation. A particular variant of this gene, found at high frequencies in Papua New Guinea and neighboring Melanesian populations, shows clear Denisovan ancestry and has been linked to reduced malaria severity, suggesting that Denisovans, who likely spent time in the tropical and subtropical regions of Asia where malaria was endemic, evolved immune variants that helped control the dangerous inflammatory responses that malaria can trigger. When modern humans moved into those same malarial regions, encountering Denisovans and interbreeding with them, some of the hybrid offspring inherited this protective variant, survived malaria infections at higher rates than those who lacked it, and passed the Denisovan gene forward through generations, spreading it through populations until it became common, reaching frequencies where it now affects the health of millions of living people who have never heard of Denisovans but carry their disease-fighting legacy in every cell. The HLA-A and HLA-B genes, which code for the proteins that display pathogen fragments on cell surfaces for immune surveillance, show particularly strong evidence of Denisovan introgression in Asian populations. Specific alleles designated HLA-A11 and HLA-B15, among others, are common in East Asia, present in substantial fractions of Chinese, Japanese, Korean, and Southeast Asian populations, and these alleles are nearly identical to sequences found in the Denisovan genome, far more similar than they are to any HLA variants in African populations, or even in the modern human sequences that gave rise to all non-African peoples. The statistical evidence overwhelmingly supports introgression rather than shared ancestry or convergent evolution. These are Denisovan genes, borrowed through interbreeding, preserved by natural selection because they worked, because they recognized and helped fight pathogens that modern human HLA variants did not handle as effectively. The geographic distribution of Denisovan immune genes creates a layered picture of archaic ancestry across Eurasia, where European populations carry primarily Neanderthal variants. East Asian populations carry both Neanderthal and Denisovan variants in different proportions, and Pacific populations carry the highest levels of Denisovan ancestry along with some Neanderthal. This stratification reflects the migration routes and encounter patterns of modern humans as they spread across the Old World, first meeting Neanderthals in the Middle East and Europe, then meeting Denisovans further east in the highlands and lowlands of Asia, with some populations experiencing multiple waves of introgression from different archaic sources at different times. The immune system, because it benefited so strongly from the adaptive variants that archaic humans had already evolved for Eurasian and tropical Asian pathogens, became the repository for this borrowed DNA. The place where archaic ancestry was not just tolerated, but actively amplified by natural selection. What emerges is a picture of modern human immune systems as fundamentally hybrid constructions, built from 3 or sometimes more ancestral sources, depending on the population. The base is Homo sapiens, the immune genes that evolved in Africa over hundreds of thousands of years, tested against African pathogens, optimized for tropical and subtropical disease environments. Layered on top, in populations that left Africa, are Neanderthal contributions, immune variants adapted to the bacterial and viral ecology of temperate and cold Eurasia, providing resistance to respiratory infections, skin bacteria, and the suite of diseases that thrive in dense populations living through harsh winters in close quarters. And layered on top of that, in Asian and Pacific populations, are Denisovan contributions, immune variants adapted to the specific pathogens of tropical and subtropical Asia, of highland and lowland environments across a vast and ecologically diverse continent, providing additional coverage, additional recognition capabilities, additional tools for fighting the endless variety of microbes that threaten human survival. Each layer represents hundreds of thousands of years of evolutionary experimentation, of natural selection testing variants against real diseases, of populations surviving or failing based on whether their immune genes could handle the threats they faced, and modern humans, by interbreeding with these archaic populations and borrowing their pre-tested solutions, compressed that evolutionary timeline dramatically. Instead of spending hundreds of thousands of years slowly evolving new immune variants through random mutation and selection, modern humans acquired functional variants instantly in a single generation through the simple mechanism of producing children with archaic partners, and natural selection then sorted through those hybrid genomes, keeping what worked and discarding what didn't, assembling immune systems that were better adapted to Eurasian environments than pure Homo sapiens could have evolved on its own in the time available. The Denisovans themselves, like the Neanderthals, eventually disappeared, their populations dwindling and finally vanishing from the archaeological record with no clear explanation for why they failed to survive when modern humans succeeded. But also like the Neanderthals, they are not entirely extinct, not completely gone, because their genes persist, still functional, still providing protection, still shaping who gets sick and who stays healthy when diseases sweep through populations. In Papua New Guinea, in Indonesia, in the Philippines, in southern China, in Aboriginal Australian communities, people carry substantial Denisovan ancestry, and much of that ancestry is concentrated in immune genes, in the HLA region and the inflammatory response pathways and the antiviral defense systems that Denisovans refined through their own deep evolutionary history, and that modern humans borrowed when they needed solutions to survival challenges they had not yet solved on their own. The Denisovan contribution is less studied than the Neanderthal contribution, less well understood, but it is no less real, no less important to the people who carry it, and as genomic medicine advances and researchers sequence more diverse populations, the full extent of what we owe to this mysterious extinct lineage will become clearer, revealing yet more ways in which ancient humans still shape modern health and survival. Today, in the age of global pandemics and personalized medicine, these ancient immune genes borrowed from extinct humans are more relevant than ever, more consequential for clinical outcomes and treatment decisions than anyone could have predicted even a decade ago. And the realization that Neanderthal and Denisovan DNA still shapes who responds to vaccines, who benefits from certain drugs, who survives infections, and who doesn't, has transformed how researchers think about human genetic diversity and forced a reckoning with the fact that medicine developed primarily using European populations, tested on genetically narrow cohorts, calibrated to immune responses that represent only one possible configuration among many, cannot simply be applied universally across all ancestry groups without accounting for the fundamental differences in immune gene variants that map onto geography and history and the specific archaic human contributions that different populations carry. The one-size-fits-all approach to medicine, already under pressure from growing evidence of pharmacogenetic variation in drug metabolism and response, faces an even deeper challenge when the variation involves not just modern human diversity, but the legacy of interbreeding with multiple extinct species whose genes are still active, still functional, still determining clinical outcomes in ways that are invisible unless you sequence genomes and look for those ancient signatures. Pharmacogenomics, the field that studies how genetic variation affects drug response, has traditionally focused on genes involved in drug metabolism, the liver enzymes that break down medications and determine how long they stay active in the body, how quickly they are eliminated, whether they build up to toxic levels or are cleared before they can work. But immune genes matter just as much for many treatments, particularly for drugs that modulate immune responses, that suppress inflammation, that enhance antiviral defenses, and here the Neanderthal and Denisovan legacy creates measurable differences in how people respond. Interferon therapy, used to treat hepatitis C and some cancers, works by ramping up the same antiviral pathways that the OAS genes regulate, and individuals who carry Neanderthal OAS variants produce different baseline levels of these antiviral proteins, respond differently to interferon treatment, show different rates of viral clearance, and different side effect profiles. Some studies have found that certain Neanderthal OAS genotypes predict better response to interferon therapy, while others predict worse response or more severe side effects, and knowing which variants a patient carries could help doctors choose optimal dosing or decide whether interferon is the best treatment option or whether alternatives should be tried first. Vaccine responses follow similar patterns because vaccines work by training the immune system to recognize and fight specific pathogens, and how well that training works depends on the genetic variants that control immune activation, antibody production, and immune memory. The HLA genes, where Neanderthal and Denisovan ancestry is so prominent, directly determine which pathogen fragments can be displayed to T cells, which means they determine whether the immune system can mount a strong response to the proteins in a vaccine. People with certain HLA types produce robust, long-lasting antibody responses to flu vaccines, while people with other HLA types produce weaker responses that fade faster, requiring booster shots or higher doses to achieve the same level of protection. The same variation affects COVID-19 vaccine responses, hepatitis B vaccine responses, essentially any vaccine that depends on the adaptive immune system recognizing and remembering specific viral or bacterial proteins, and some of that variation traces back to archaic human ancestry, to HLA alleles that Neanderthals or Denisovans carried, and that modern humans borrowed and still use to recognize pathogens that did not exist when those archaic humans walked the earth. The COVID-19 pandemic forced this issue into stark visibility, turning the entire global population into an unintended natural experiment on how ancient immune genes affect modern disease outcomes. When researchers scrambled to understand why severity varied so wildly, why some countries and some demographic groups were hit harder than others, even after accounting for age and health conditions and healthcare access, genetic studies kept pointing to the same regions of the genome. Chromosome 3 with its Neanderthal OAS cluster that protects against severe disease. Chromosome 12 with its Neanderthal inflammatory variants that increase risk. The HLA region with its complex mixture of modern human, Neanderthal, and Denisovan alleles that affect how quickly and how strongly the immune system responds to the virus. The protective Neanderthal haplotype on chromosome 3, the one that enhances antiviral responses, is present in about 30% of people in East Asia, particularly in China and Japan, but only about 8 to 10% in Europe, a distribution that might partly explain why, even accounting for public health measures and other factors, East Asian countries showed different patterns of severe disease than European countries in the early waves of the pandemic before vaccines were available. The risk-increasing Neanderthal variants on chromosome 12, the ones that promote excessive inflammation, show the opposite geographic pattern, more common in South Asia and Europe than in East Asia, and studies found that people who carried 2 copies of the risk haplotype were roughly twice as likely to require hospitalization if infected compared to people who carried 0 copies, a substantial effect that operated independently of age, sex, or underlying health conditions. This means that part of who lived and who died during COVID-19, part of who ended up on ventilators and who recovered at home with mild symptoms, was determined by genetic variants inherited from Neanderthals who had been extinct for 30,000 years, who never encountered anything resembling a coronavirus, whose immune genes nonetheless shaped outcomes because evolution is not specific, because the same variants that helped fight Ice Age infections also affect how the body responds to modern pathogens, sometimes helpfully and sometimes harmfully, depending on the specific disease and the specific immune mechanisms it triggers. Precision medicine, the effort to tailor treatments to individual patients based on their specific genetic makeup, must grapple with this reality, must incorporate archaic ancestry into risk prediction models, treatment algorithms, and clinical decision-making. A person of East Asian descent with high Denisovan ancestry in immune genes might respond differently to immunotherapy for cancer than a person of European descent with high Neanderthal ancestry, not because of race in any social or cultural sense, but because they carry different functional variants in the genes that control immune activation, inflammation, and the ability to recognize and attack tumor cells. A drug that works brilliantly in clinical trials conducted primarily on European populations might work less well or cause more side effects in Asian or Pacific populations who carry different archaic immune variants, and failing to account for this variation means some patients will be undertreated, others overtreated, and the precision promised by personalized medicine will remain elusive for anyone whose genetic background was not represented in the original research. The implications extend to infectious disease surveillance and pandemic preparedness as well. If we know that certain Neanderthal or Denisovan immune variants affect susceptibility to viral infections, affect severity of inflammatory responses, affect who can clear an infection quickly, and who develops chronic disease, then we can use genetic data to identify high-risk populations before a pandemic hits, to target vaccines and treatments more effectively, to understand why outbreaks spread differently in different regions. We can develop drugs that work with, rather than against, the specific immune responses that different genetic backgrounds predispose toward, designing treatments that enhance the antiviral pathways that Neanderthal OAS variants already boost, or that dampen the excessive inflammation that Neanderthal TLR variants trigger, calibrating interventions to the actual immune systems people have, rather than to some idealized average that represents no one accurately. This also raises profound questions about gene therapy and genetic enhancement, about whether we should attempt to edit out harmful archaic variants while keeping beneficial ones, about whether future medicine will involve not just treating diseases, but optimizing genomes, selecting which ancient legacies to preserve and which to discard. CRISPR and other gene editing technologies now make it theoretically possible to remove the Neanderthal variants that increase autoimmune disease risk while leaving the variants that protect against viral infections, to engineer immune systems that have all the benefits of archaic introgression without the costs. But this is ethically and practically complex because we do not fully understand all the effects of these variants, because the same gene variant might be protective in one context and harmful in another, because evolution has had 50,000 years to test these combinations, and we have had only a few decades to study them. And hubris about our ability to improve on evolutionary solutions has led to disasters before when we thought we understood systems better than we did. The Neanderthal and Denisovan immune genes also complicate efforts to create universal vaccines, treatments that would work equally well in all populations regardless of ancestry. If different populations carry fundamentally different immune gene variants that respond differently to the same antigens, then a vaccine optimized for European immune systems might be suboptimal for Asian or African immune systems, and truly universal protection might require multiple vaccine formulations or adjuvants tailored to work with the specific HLA types and inflammatory pathways that different populations carry. This is not a reason to abandon universal health goals, but it is a reality that must be acknowledged and addressed, a consequence of human evolutionary history that creates biological diversity significant enough to affect medical outcomes. Diversity that is not trivial or cosmetic, but functional, written in the genes that control life and death responses to pathogens. The ancient humans who contributed these genes did not intend to create these challenges for modern medicine, could not have anticipated a world where their genetic legacy would complicate vaccine development or drug approval or clinical trial design. But their inadvertent gift of immune diversity is the reality we must work with. The hand that evolution dealt us through the contingent accidents of interbreeding and selection and survival 50,000 years ago, in a world that no longer exists, but whose consequences still echo through every clinic and hospital and pharmaceutical development program, trying to keep humans healthy in the 21st century. The Neanderthal story, the Denisovan story, the entire saga of archaic human DNA still living inside billions of people and still controlling immune responses to modern diseases, is really a story about what it means to be human, about extinction that isn't quite extinction, about boundaries between species that turn out to be permeable rather than absolute, about the ghosts we carry and the futures they shape in ways that force us to reconsider fundamental questions of identity, ancestry, and what we owe to relatives so distant that they are classified as different species, but so intimate that their genes still function in our cells, still protect us, still make the difference between sickness and health, between surviving a pandemic and succumbing to it. When we say that Neanderthals are extinct, we mean that there are no living individuals, no communities that maintain Neanderthal culture or language or identity, no populations that can be pointed to and identified as Neanderthal rather than modern human. But genetic extinction is something different, and by that measure, Neanderthals are very much not extinct, are in fact among the most successful human lineages ever to exist, with their genes distributed across half the planet, present in several billion people, still active and functional after 50,000 years, still shaping biology in consequential ways. They did not disappear. They dissolved, their genetic material absorbed into the much larger modern human population, their distinct identity lost, but their adaptive innovations preserved wherever those innovations provided value. This challenges the traditional concept of extinction as an endpoint, a final vanishing, a closing of an evolutionary branch that leaves nothing behind. The passenger pigeon is extinct in this absolute sense, gone completely, with no descendants and no genetic legacy in any living species. The dodo is extinct in this sense, as are the woolly mammoth and the saber-toothed cat, and countless other species that left fossils but no living inheritors. But Neanderthals and Denisovans occupy a different category, extinct as recognizable populations but living as distributed genetic elements, as sequences embedded in other genomes, contributing to other species' survival, achieving a kind of immortality through hybridization that is perhaps more durable than survival as a pure lineage, because it spreads the risk across billions of bodies rather than concentrating it in a single isolated population vulnerable to environmental catastrophe or demographic collapse. The Neanderthals as a distinct people are gone, but Neanderthal immune genes have never been more numerous or more widespread than they are today, carried by billions of people who might never think about their ancestry, but who owe part of their ability to fight infections to people who looked different, lived differently, and disappeared as a recognizable group tens of thousands of years ago. This realization undermines the old narrative of human evolution as a story of progress and replacement, of superior modern humans outcompeting inferior archaic humans through better brains or better tools or better social organization, driving them to extinction and taking over the world as the single victorious lineage. The genetic evidence reveals a messier, more collaborative story, where modern humans succeeded not by being pure or superior, but by being hybrid, by borrowing adaptive innovations from the populations they encountered, by incorporating archaic genes that solved problems modern humans had not yet solved, by stealing, essentially, hundreds of thousands of years of evolutionary experimentation that had already been tested by natural selection in the environments modern humans were trying to colonize. The success of modern humans is partly a success of assimilation, of genetic theft, of hybrid vigor, where the combination of different lineages produced offspring better adapted than either parent population alone, and that success depended critically on the contributions of species we now call extinct, but whose genes were essential to our survival. Hybrid vigor is a well-known phenomenon in biology, where crossing two genetically distinct populations produces offspring with better survival, faster growth, higher fertility than either parent line, the result of combining different adaptive variants, masking harmful recessive mutations, and bringing together innovations that evolved independently in response to different challenges. Agriculture has exploited hybrid vigor for thousands of years, crossing different strains of crops and livestock to produce varieties that yield more, resist diseases better, tolerate stress more effectively than purebred lines. The modern human genome is, in a sense, the product of natural hybrid vigor, the result of interbreeding between lineages that had been separated long enough to accumulate different adaptive solutions, but not so long that they were reproductively incompatible, and the immune system benefited most clearly from this hybridization because it is where the different lineages had evolved the most obviously complementary innovations, where Neanderthal variants provided tools for fighting Eurasian pathogens and Denisovan variants provided tools for fighting Asian tropical pathogens, and modern human variants provided tools for fighting African pathogens, and the hybrid offspring that inherited useful variants from multiple sources had more comprehensive immune defenses than any pure lineage could have evolved in the time available. This genetic diversity, this patchwork inheritance from multiple ancestral sources, is not a historical curiosity but an ongoing survival advantage, because pathogens evolve, new diseases emerge, and a population where everyone has identical immune genes would be devastatingly vulnerable to any pathogen that happened to evade that particular configuration. The influenza pandemic of 1918 killed tens of millions of people, but it did not kill everyone, did not drive humanity extinct, because human immune diversity meant that some people carrying certain HLA types or certain TLR variants or certain combinations of immune genes were able to mount effective responses and survive, and those survivors repopulated the world, carrying forward the variants that worked against that particular pathogen. COVID-19 followed the same pattern, killing millions but not billions, severe in some people and mild in others, and part of that variation traced to Neanderthal immune genes, to ancient variants that happened to provide protection or happened to increase risk in ways that no one could have predicted, but that ensured the species survived because not everyone responded the same way. Because genetic diversity is resilience, is insurance against catastrophic loss, is the reason why we are still here after countless plagues and pandemics that would have wiped out a genetically uniform population. The future of this legacy is uncertain and raises questions that are as much ethical and philosophical as medical or scientific. We now have the technological capability to edit human genes, to use CRISPR and other tools to remove variants we consider harmful and insert variants we consider beneficial, to engineer immune systems according to our current understanding of what works best, rather than accepting the inheritance that evolution and history have handed us. We could, in principle, screen embryos or edit germline cells to eliminate Neanderthal variants that increase autoimmune disease risk, to remove the TLR alleles that cause excessive inflammation, to select against the chromosome 12 haplotype that increased COVID-19 severity. We could select for the protective variants, the chromosome 3 OAS cluster that helps clear viral infections, the HLA alleles that recognize broad ranges of pathogens, building optimized immune systems that have all the benefits of archaic introgression without the costs. This is not science fiction, but near-term possibility, technologies that already exist and are being refined that will inevitably be applied to human reproduction as they become safer and more precise and more socially acceptable. But the wisdom of doing so is far from clear, because evolution is smarter than we are in the sense that it has tested these variants across millions of individuals over thousands of generations in real environments against real pathogens, and we have studied them for a few decades in artificial contexts that may not capture all their effects, all their interactions, all the ways they matter in the complex biological reality of a human lifetime. The Neanderthal variants that increase autoimmune risk in the modern low-pathogen environment might be life-saving in a different environment during a different pandemic against a different pathogen that we have not encountered yet and cannot predict. Diversity itself has value, insurance value, option value, the value of not putting all our evolutionary eggs in one basket, and homogenizing human immune systems towards some current optimum might reduce that diversity to dangerously low levels, leaving us vulnerable to threats we cannot anticipate. The Neanderthals and Denisovans did not plan to provide us with genetic tools for the future. They simply lived and adapted and died, and the variants they evolved were preserved by natural selection because they worked in the contexts where they were tested. And some of those variants still work today, and others might work again in future contexts we cannot foresee, and eliminating them might be a mistake we only recognize when it is too late, when some new pathogen emerges and we discover that the ancient variant we edited out was the one that would have saved us. The question of identity, of what makes us human, becomes more complex when we acknowledge that we are not purely one species, not a single evolutionary branch that outcompeted all others, but a braided stream, a combination of lineages that merged and mixed and produced something that is greater than any component alone. The people who contributed to your genome include anatomically modern Homo sapiens from Africa, Neanderthals from Europe or Western Asia, possibly Denisovans from Eastern Asia, and potentially other archaic human lineages we have not yet identified, but whose genetic traces might be hiding in populations that have not been thoroughly studied. You are not purely Homo sapiens if you have non-African ancestry. You are a mosaic, carrying genes from multiple species, and those genes are not historical relics, but active contributors to your biology, shaping how you look, how you fight disease, how you respond to environments. This does not make you less human, but it expands what human means, makes it a broader category that includes not just our direct ancestors, but the genetic contributions of related species that we absorbed along the way, species that gave us tools we needed and in return achieved a kind of genetic immortality, their sequences preserved and propagated far beyond what they could have achieved as isolated populations. The practical implications ripple through medicine, through public health, through how we think about disease prevention and treatment, and the development of drugs and vaccines. Recognizing that human immune diversity is not trivial, not a minor variation around a single theme, but a fundamental difference reflecting contributions from multiple extinct species adapted to different environments, means that universal one-size-fits-all medical approaches will always be imperfect, will always work better for some populations than others, unless we explicitly account for archaic ancestry and design treatments that accommodate the genetic diversity that history has created. It means that studying diverse populations is not optional, not a matter of inclusion for its own sake, but scientifically necessary to understand the full range of human immune function and to develop interventions that work across all the genetic configurations that exist in our species. It means that the extinction of Neanderthals and Denisovans is not a closed chapter, not something that happened in the distant past with no relevance to the present, but an ongoing influence that shapes clinical outcomes today and will continue shaping them for as long as humans carry archaic genes, which may be forever, or at least until we make deliberate choices to edit them out and accept the consequences, whatever they turn out to be. So as you drift toward sleep, as the day's concerns fade and your mind settles into rest, think about the ancient humans breathing through you, fighting infections in your cells, producing proteins that recognize pathogens and coordinate defenses, contributing in ways large and small to your survival, to your ability to wake up healthy tomorrow and the day after. They are not separate from you, not foreign DNA invading from outside, but part of what you are, woven into your biology at the most fundamental level, as much a part of your inheritance as the genes you received from your recent ancestors, from your parents and grandparents, stretching back through recorded history. The Neanderthals who contributed those genes never knew you would exist, never imagined a world of cities and airplanes and global pandemics, never guessed that their immune systems would still be working tens of thousands of years after they died, protecting descendants they could not have conceived of, solving problems in environments they never encountered, achieving a form of immortality stranger and more distributed than any myth or religion ever proposed. They are extinct, and they are not extinct, gone and still here, disappeared from the earth as living populations, but present in billions of bodies, still relevant, still functional, still making the difference between health and sickness, between survival and death, ghosts that never left, ancestors who never stopped protecting their descendants, even descendants who belong to a different species, even descendants who are still trying to understand what it means that extinction is not always final, that species boundaries are not always absolute, and that the ancient humans whose bones crumble in caves and whose tools rust in museum cases are still with us, invisible but essential, silent but active, extinct everywhere except in the code that builds and maintains your body, where they live on, fighting the same ancient war against disease and death that every living thing has fought since life began and will go on fighting as long as you do, as long as any of their hybrid descendants survive, carrying forward this strange, complicated, magnificent legacy of interbreeding and survival, of extinction that became incorporation, of death that became immortality through the only mechanism that truly lasts. Genes passed forward, generation after generation, still working, still mattering, still keeping you alive tonight and every night. These ghosts still breathing, still protecting, still here.